Hello everyone, welcome back to another session of regeneration biology, and in today's class we will learn about types of adult stem cells and the trans-differentiation for tissue engineering. What is their potential of trans-differentiation and tissue engineering? So we will learn about what are the pros and cons, what are the different angles of trans-differentiation to be taken care of. So adult stem cells are undifferentiated cells found in various tissues after birth that can self-renew and differentiate into specialized cell types. We know that because adult stem cell stays throughout the lifespan of an organism. We have seen this multiple times. We will see today's class also very quickly. We'll flash through them. They are classified based on their differentiation potential, including the pluripotent and multipotent stem cells. Pluripotent stem cells, such as embryonic stem cells and induced pluripotent stem cells, can differentiate into all cell types present in the body. They can give rise to each and every. We have more than 100 different cell types in our body, so it can give rise to any of them. Multipotent stem cells, like the mesenchymal stem cells, short form known as MSCs, can differentiate into more limited range of cell types within a specific tissue. Say bone marrow stem cells, they are multipotent. They can give rise to only RBC, WBCs and some of the immune cells like natural killer cells, blood platelets, macrophages, etc. This is an example. So you have bone and you have bone marrow, and inside the bone marrow you have got the haemopoietic stem cell, and it gives rise to two lineage. One is a lymphoid progenitor cell. So this haematopoietic stem cell gives rise to multipotent cell, which is give rise to lymphoid lineage and myeloid lineage. Lymphoid lineage give rise to natural killer cells, T lymphocytes and B lymphocyte. Whereas myeloid progenitor cells, they can give rise to neutrophil, basophil, eosinophil and monocyte, macrophage, platelets, RBCs, etc. And these stromal cells, they can give rise to bone or cartilage, and also there is adipocytes that is present, and it can give rise to fats storing cells. They can be given rise to, and also we can easily tweak these cells by trans-differentiation into sometimes the skeletal muscle cell or hepatocyte of the liver, etc. And some situations you can make them into bone itself, and also it can be made into other stem cell, hematopoietic stem cells itself, and also some other niche type or some unique type of cells also can be made. So multipotent cells found in the bone marrow, they usually give rise to the bloodstream cells, but they differentiate into in a systematic manner, and which will be released into the bloodstream, and that is the routine scenario. But can this rule be violated? That is what we will try to understand. So types of adult stem cells and their hierarchies, we'll quickly see. The principles of renewing tissues. Stem cells are able to make a copy of themselves. They can self-renew, divide rarely and high potency rate, and they are found rarely, and they give rise to a committed progenitors. That means they have a defined direction now. They are called committed progenitors. They are transiently amplifying cells. They are multipotent. They divide very rapidly, and they have no self-renewal capability. They must come from the stem cell, and they give rise to specialized cells. So this is the simple principle of renewing of the tissue. And hematopoietic stem cells, we have already seen it, and this is another description that hematopoietic stem cells of the bone marrow, they give rise to different steps like NK cell, T cell, B cell, dendritic cell, etc., etc. Now, the mesenchymal stem cell, which is basically derived from the transformation of a epithelial characteristic, which is a static in nature, whereas once it becomes mesenchymal, it becomes more mobile in nature. So MSCs can make copy of themselves, and once they reach committed progenitors, they can give rise to bone, cartilage and fat cells. They can give rise to these cells. And natural neural stem cell. Neural stem cells, also known as NSCs, they are derived from the stem cell and give rise to committed progenitors and give rise to various neurons, interneurons, oligodendrocytes, type 2 astrocytes, type 1 astrocytes, etc. So gut stem cells, this is another type of adult stem cells. They can make a copy of themselves and they give rise to committed progenitors, and they can give rise to Paneth cells, goblet cells, endocrine cells, columnar cells, etc., which is specialized cells. So we are trying to understand, can these cells be pushed into any cells of your liking? And there is one more source of cell which we can make is, which is called induced pluripotent cell. That is, any cell in the body you can pick it up and convert into a pluripotent cells. That means it is not an adult stem cell, but rather you can make stem cell from an adult any cell or random cell. So induced pluripotent stem cells, they are made by the genetic reprogramming. We have seen it, Yamanaka factor, Thomson factors, etc. And once you have the iPSCs, you can culture them on a layer and you force them to differentiate into any cell type. So in this way, we can convert a iPS cell into any cell type of your choice, and that can be used. And this picture shows that a normal cell you took and you converted them into stem cell, and now you convert the stem cell into any cell type of your choice. And let us see what is trans-differentiation. Trans-differentiation is a very interesting concept, that certain adult stem cell types can differentiate into cell types seen in organs or tissues other than those expected from cells of the predicted lineage. This reported phenomenon is called trans-differentiation. In simplistic sense, I can tell you, say bone marrow stem cells are giving rise to all blood related cell, what we have seen it. Now, the bone marrow stem cell, can you make them make a neuron? Can you make them make a liver cell? Can you make them make a muscle cell? So this is trans-differentiation. Actually, they won't make that, but you can coax them to make it. So example, brain stem cells that differentiate into blood cells, or blood forming cells that differentiate into cardiac muscle cells. Many such examples can be thought of. So this is the whole concept of trans-differentiation. So what is the role of trans-differentiation in tissue engineering? This is something which we have to look into. Adult stem cells are clonogenic, self-renewing and pluripotent cells with a plasticity to differentiate into types of particular tissue in which they reside, and often to trans-differentiate into different types of tissues. This property is being made use of in tissue engineering. In the sense, it is very easy to collect bone marrow stem cells from a patient. If the patient do not have any genetic issue, you can happily collect the bone marrow cells. Once you collect it, you can program the environment of that cell. You don't need to change the genome of anything. The living conditions of that cell can be changed, and you can now get any cell of your choice. If you want kidney cell, you can make it. If you want heart cell, you can make heart cell, like that. So which is one of the easiest and less cumbersome and cost-effective method. So trans-differentiation, also known as metaplasia. In cancer, you may have heard this as a disease association. Metaplasia basically means standing far away. That is what metastasis. Stasis means standing, meta means other place. That is the meaning of it. Normally, say someone have a cancer in the liver, and now this liver cancer cell went and formed a colony in the brain. And that is usually we refer to as metastasis or a cancer's progression. This is trans-differentiation is somewhat related, but it is called metaplasia. So here what refers to converting one type of cells to another. It is nothing to do with any cancer formation or something. Metaplasia basically means you are converting one cell's property to another. How this happens, an existing differentiated cell also can get converted into another differentiated cell. Not by direct jumping, it will go back one step backwards, and from that step it will give rise to another cell type. So a muscle cell went back to its progenitor. Now instead of becoming muscle cell, it is now becoming liver cell, something like that. Involving switching of stem cells into a different cell type is often referred to as metaplasia. In tissue engineering, trans-differentiation induces stem cells to differentiate into specific cell types needed for tissue repair or regeneration. So this is the sole purpose of using trans-differentiation in tissue engineering. That is, can induce the stem cells to differentiate not into its natural course of differentiation, into a different course of differentiation. For example, MSCs, mesenchymal stem cells, can be induced to differentiate into bone, cartilage or muscle cells for tissue engineering applications. So if you have MSCs present from an organism, I can make, I need a bone, I need a muscle cell, so I can make as my liking. So let us see some examples of trans-differentiation in tissue engineering. Bone tissue engineering, MSCs can be induced to differentiate into osteoblast, that is bone forming cells, or used for bone regeneration. Cartilage tissue engineering, that is MSCs, can be induced to differentiate into chondrocytes, that is cartilage cells, for cartilage repair. Like your ear pinna is cartilage, your nose, many part of your body is having cartilage is still there, and you can use cartilage synthesis using MSCs. Muscle tissue engineering, MSCs can be induced to differentiate into myoblast, that is muscle forming cells, for muscle regeneration. Skin tissue engineering, skin stem cells can be induced to differentiate into keratinocytes for skin regeneration. So the list continues. Don't think that there is only 4 types of cells can be produced. Some examples are mentioned here. And let us understand what is the growth and isolation of stem cells and test to confirm the stem cells presence. So what are the ways in which people approach? The trans-differentiation potential of adult stem cells and their capacity for tissue renewal and damage repair have attracted too much attention from biotechnologists and clinicians, because trans-differentiation, like I told you, collecting bone marrow cell is very easy, and there is a plenty of stem cells out there. Of course, you can get a stem cells from skin, or you can get from intestine, wherever you want you can collect it. But bone marrow stem cell collection, we have lot of bone marrow throughout your body, so tissue is not in short supply, so you can easily collect it. People even do donate, like someone who have a leukemia, you can take from the sibling or the genotypically closer individual, you can take the bone marrow and you can even donate to a recipient. So it is very less problematic. Stem cell isolation and in vitro maintenance are important for applied application of this technology, or in the for implementing into an applied biology field. Take the stem cell and you should be able to culture them without losing their property. And how do you isolate a stem cell? When you collect bone marrow, you cannot put one needle and take the cell and tell that, okay, now I collected all the stem cell. It will be a mixture of cells. So you should be able to isolate the stem cell from that. Flow cytometric separations of stem cells or positive and negative selections using magnetic beads tagged with antibodies targeting specific markers on the surface of stem cells are used routinely now in many such applications. So you can have a mixture of, say I have 100 cells, in that only 5 are stem cells. I will be able to identify that 5 making use of the marker. Alternative approaches of stem cell identification have also been proposed based on specific behavior of individual stem cell. Every stem cell have got a unique set of marker, and if you want to identify that stem cell from a group of cell, you should go after those markers which is unique of that particular cell. One of these approaches exploits stem cell homing characteristics. Homing characteristics means its identity is retained in this homing or the marker genes. These and other principles effectively isolated stem cells for research and biological applications. Scientists do based on these approaches. We will see one example, though there are multiple methods. One prevalent approach is isolation of stem cells by flow cytometry. Flow cytometry passes mixture of cells tagged by appropriate fluorochrome labeled stem cell markers through a laser beam. So stem cell has got a marker, and now you are putting an antibody against that marker, and that antibody is having a fluorophore. So antibody will not bind to every cell, it will bind only to that cell which has got this marker expressed. And this marker bearing cell is nothing but the stem cell which you are hunting for. Now, you have 100 cells or maybe 1000 cells. In that, maybe 10 cells are stem cells which you are looking for, and this antibody go unbound. And now, this antibody has got a fluorophore. Let's say fluorescent tag. The cells scatter the fluorescence, which provides information on the cell's morphology, composition of surface proteins, DNA content, and cytoplasmic processes. So you will be able to measure lots of properties of the cells based on the scattering, fluorescent scattering, which is present, which is derived from the fluorophore attached onto the antibody, which already bound to the functional marker present on the surface of this stem cell. So flow cytometry's advantage is the flow speed, which allows quick processing of a large population of cells. The quick processing of a large population of cells is possible only if you have the flow cytometric approach, if you adopt for separating the cells. The cytometric analysis goes one step further and allows cell sorting, which breaks the fluid stream containing the cells into droplets by piezoelectric perturbation. So this is the technology that is applied. The cytometric analysis can be further tweaked to refine it, which is also leading to the cell sorting, which breaks the fluid stream containing the cells into droplets by piezoelectric perturbation. Slight electric charges will be applied. Accordingly, it will have a different behavior. It is then possible to detect a selected droplet with the precise timing on a charge, and this droplet has charge which can be detected given to the stream as it passes through an electric field. So the cells can be separated at a given electric charge, and the droplets containing the stem cells, every cell is present in a given droplet, and that droplet has got stem cells also, and this stem cells of interest bearing droplets are deflected in the electric field, which can be collected, and you can culture them or you observe them, etc. So now what you do for the growth and validation of stem cells presence? Like other mammalian cells, stem cells are grown and maintained in 37 degrees Celsius in humidified cell culture incubators under a 5% CO2 atmosphere. Like any other cell culture, stem cells also need same kind of environment. The media requirements vary among individual stem cell types, and it is important that not to induce differentiation of the cultured stem cell. Like we have seen it, there will be feeder layer, leukemia inhibitory factor, etc. has to be put in the medium so that they will not differentiate. Stem cells are very easy to differentiate. If you like, there is a joke people say, if you take a stem cell petri dish and simply stare at it, it will differentiate. You don't need to do anything. It is that sensitive. It will not die. It is itching. It is waiting to differentiate. You take a petri dish, stem cell petri dish, keep on your bench for 10 minutes and put it back. Because your bench is not at 37 degree, it is in room temperature, enough. They would have become neurons or some other cell type. So it is almost like you have been given a soap bubble, and I am telling next 5 hours, the soap bubble should not break. It is that sensitive. Cell will not die, but cell will differentiate. So maintaining the stemness or maintaining stem cell as stem cell is the challenge. But growing, culturing, etc. is easy if you follow the protocol as it should be. Any insult you provide, it can differentiate. HSCs are obtained from the bone marrow, aspirates, placental or umbilical cord blood, and their high growth rate makes them prone to differentiation in culture. This is a challenge which you should be wary of. Bone marrow origin stem cells are grown in culture media supplemented with serum, while HSCs usually require a co-culture system hematopoietic stem cells require a co-culturing system containing fibroblast feeder layers that support the growth and differentiation. Normally, it supports the growth and differentiation when required. So you have to give, sometimes it may be changing the serum concentration, sometimes you are turning on certain genes or inducing some growth factors, etc. Differentiation is very much possible. So the change of cytokine conditions in such culture systems affects the differentiation of stem cell. Any cytokine concentration you affect either concentration or putting a new cytokine, any change can have a huge implication in the differentiation of stem cell. The effects of specific culture conditions and differentiation speed vary among different stem cell populations. It keeps varying. So bone marrow stromal cells attach to culture dishes and grow slowly for weeks before differentiation. First, the stem cell should increase in number and then they will tend to differentiate. Neural stem cells from fetal or adult brain tissue can grow suspended in a culture medium without additional serum supplements. This depends on neural stem cell. If every stem cell have got its own unique properties, unique niche, unique pros and cons, etc. So neural stem cells are like that, and from which if you isolate from fetal or adult brain tissue can grow in suspended culture medium, which is something unique. Why it is again mentioned is normally cells will grow in attachment only, but they can grow in suspended culture medium without any specific serum supplement. And then comes the validation of these cells. Validation of the presence of stem cells in a culture system with histochemical methods, antigenic markers, and morphogenetic studies primarily depends on the surface morphology and immunophenotype of the stem cells of interest. Like when you say bone marrow stem cells, or when you say kidney stem cell, or when you say heart stem cell or liver stem cell, each of them have the potential to give rise to the respective cell types. However, sometimes, as you saw in the previous slide, stem cell and there is a committed progenitor. So what is the difference between stem cell and the committed progenitor? The difference will be reflected in their surface marker. So based on the markers, you can identify, is it a committed progenitor or is it a core stem cell? So that is why the markers come into picture. These are made use in their validation. For in vitro studies, cytogenetic analysis or RT-PCR, reverse transcription of by quantitative PCR, reverse transcription and then do the PCR. RT-PCR based methods are commonly applied. So let us see adult stem cell plasticity. Adult stem cell plasticity and the implications in regenerative medicine. Cell-based therapy may soon represent a new strategy to treat a wide array of clinical conditions. It is very, very promising. Using adult stem cells instead of human embryonic stem cells for therapy avoids all kinds of ethical problems because you are not damaging any embryo, no questions will be asked. It has two additional advantages. Advantage A, adult stem cells can be isolated from patients, and this overcomes the problem of immunological rejection because it is from self. There is no question of non-self. The advantage B, the risk of tumor formation is greatly reduced as compared to the use of embryonic stem cell. Because embryonic stem cells, even if they are iPSC cells, they are tweaked. But when you take bone marrow stem cell, when you take your own adult cell, it is from you. It is normally present in you. There is nothing great modification is happening. While pluripotency and plasticity are considered properties of early embryonic stem cell, adult stem cells are traditionally thought to be restricted in their differentiation potential to the progeny of a tissue which they reside. Means they are already tamed, properly tamed cells. When parts of an organ or tissue are transplanted to a new site, the transplanted tissue maintains its original character. So this is one good advantage that this trans-differentiation potential when you use it. Similarly, when dissociated cells from an organ or tissue are cultured, they also tend to maintain their original phenotype despite losing some differentiation properties. They do not acquire differentiated characteristics of a different cell lineage normally. They can be forced to acquire a different cell characteristics. Normally, it will retain its normalcy. So this is the diagram that is illustrating the plasticity of bone marrow derived cells. Like you have bone marrow derived cells, and this arrow, direct arrow, indicates fusion, second indicates true trans-differentiation. Fusion means you are taking the stem cell and putting in an environment, and you're expecting the stem cells to become that tissue. Like you are an Indian and I put you in Rome and I'm expecting you to become like Romans. Or other option is, while in India itself, I train you how to behave and work like Romans and just putting you. So that is trans-differentiation. So normally the hematopoietic stem cells and mesenchymal stromal cells, they are vulnerable for both. Vulnerable in the sense they are amenable for both. That is, they can be fused or they can be trans-differentiated. And they can become skeletal myocytes, cardiomyocytes, pancreatic beta cells, hepatocytes, neural and glial cells. That means depending upon the need or depending upon the decision taken by the researcher or the clinicians, you can adopt either dump these cells into the tissue, this hematopoietic stem cells or mesenchymal stromal cells, you put it into a tissue and expect these cells to become that, or in a petri dish you convert them into liver cell or cardiomyocytes and then put it into the respective tissues. So this is a diagram illustrating the different mechanisms of cell fate switching in adult stem cell. So this diagram, you can see how the starting material, it becomes terminally differentiated cell it becomes, and the terminal differentiation can come from either trans-differentiation, or it can come from de-differentiation, or it can come by cell fusion. So you have one cell, this is a stem cell, and it became, say if I name this as A and it became B. B is a terminally differentiated, but that is done via trans-differentiation. But then there is one same cell, it can have an intermediate progenitor cell and it gives rise to cell A and cell B, two different cell types, and this is called a de-differentiation. And this happens in the retina. The Muller glia de-differentiate and then give rise to the rest of the neurons. It's not trans-differentiation. So here what happens, this existing stem cell now fused with another cell, means a cytoplasmic mixing happened and you end up getting a heterokaryon. And this heterokaryon now gives rise to the nuclear mixing and resolution happens and you end up getting a stable heterokaryon polyploid and you end up getting a cell fusion and you end up getting a unique fate. So these are all the different mechanism of fate switching. All are trans-differentiation only. In the net outcome is trans-differentiation only the route is different. So what are the challenges and future directions? Although having said that, it's a very promising approach, trans-differentiation is excellent, etc. Some troubles, like any other system, it also lurks. Ensuring trans-differentiated cells long-term stability and functionality is a major challenge in tissue engineering. Okay, I put the bone marrow stem cells and put it into the liver and liver is fine. Will it stay like that forever or will it start showing its original color? This is one question. Factors like epigenetic memory and incomplete conversion can affect the stability and function of trans-differentiated cell. Say I took my bone marrow cells and put it into my brain. It made neuron and it was all fine. Now will it switch back into bone marrow or some other cell type? Because it started memorizing its old lineage. This is one trouble. Further research is needed to understand these factors and developing strategies to improve trans-differentiated cells long-term stability and functionality in tissue engineering application. So the promise is there, but time testing you cannot do overnight. Time testing only when you see, oh, it stayed for several decades, then we can get the true benefit out of it. So these are all the points one has to keep in mind while considering trans-differentiation as a stable option of fixing a damaged tissue. So we will learn more about the regeneration biology in the next class. Thank you.